In this video, I will be discussing how the orientation of microtubules are organized or achieved in the cell. So take note, microtubules are actually polar in nature. There is a positive end and then there is a negative end. So because of this polar nature of microtubules, they may be used by certain motor proteins like the kinesin, which can move from the negative to the positive direction, and dyneins, which can move from positive to the negative direction. And these motor proteins are the ones that carry with them the vesicles or the organelles that need to be moved inside the cell cytoplasm. So again, we are going to find out in this discussion how the orientation of microtubules is achieved in the cell. And that is because of an organizing center. So we call as the microtubule organizing center, or the MTOC. And the most important of these is actually the centrosome. Okay? So the centrosomes appear like this in drawings of cells. So if you've been drawing cells and then their parts, I'm sure you've drawn the centrosome. So it's composed of two centrioles, as you can see here, that are arranged sort of in right angle of each other. So here we have one, and here we have another. And then they're surrounded by a dense material, but there's no membrane. This material is called the pericentriolar material. And then if we look at the structure of each of these centrioles, we have 1, 2, 3, 4, 5, 6, 7, 8, 9 of these portions with 9 spokes. And we can see here that each of these are made of 3 tubules, microtubules. You have the C, you have the B, and you have the A. So only the A is a complete microtubule. B and C are sort of half in diameter. If you can see the cross-section of these microtubules. So here are pictures taken using an electron microscope of the centriole. So the centriole is found in the centrosome. And here we can see that the centrosome is indeed where all of the microtubules lead to, or where they are all connected. So it is indeed really the organizing center of the microtubules. So how are the microtubules arranged with respect to the centrosome? So here we can see the centriole. And the first part, or the tip of the microtubule that's towards the centrosome, is the negative end. And the positive ends are the ones that are going away from the centrosome. So the building up or the growing of microtubules is going outwards because that's where the positive ends are. Okay, that's where the beta tubulins are located. So we can see here the elongation is going towards away from the centrosome. And there are experiments when a cell, for example, like this melanophore, is divided into 2. And then only this bigger portion as the organizing center, the smaller portion lost the organizing center, but still has the microtubules. What actually happens is that the microtubules will organize themselves even with the absence of the original centrosome, as you can see here. So there is really an inherent characteristic of organizing the microtubules. So in different cells, there's a different way of organizing or arranging the microtubules. In spherical cells, the centrosome is usually located near the center, and the microtubules move away from the centrosome. So this is the negative end near the center and the positive end near the periphery of the cell. In columnar epithelial cells, the centrosomes are located usually near the top of the cell, and the microtubules are in a direction from the top going downwards. And then there are those that are not associated with the centrosome, but still the direction is from the top towards the basal lamina. And then for neurons, because the axon is quite thin, the centrosome is located in the cell body instead. And then there is an organization of microtubules in the cell body, as shown here, from the center to the periphery. And then there are microtubules that are not associated with the centrosome, running along the length of the axon, with positive here near the cell body and negative near the cell body and positive at the tip of the axon. Now take note, certain animal cells like the mouse oocyte lacks centrosomes, and plant cells do not have any obvious microtubule organizing centers at all. They don't have centrosomes, nor centrioles, nor any other obvious alternative for the centrosome, the organizing center. Then, for another type of microtubule organizing centers would be found in cells with flagella. So, in this spherical cell, we have here the centrosome, if you can remember. That's the organizing center. So this is an example of a secondary spermatocyte wherein the cell is still spherical in shape, but as it transforms into a spermatogonium or a mature sperm cell, the centriole and the centrosomes are gonna be moved to become this structure we call as basal body. And they will be the origin of the microtubules that would make up the flagella or flagellum of the sperm cell. So let's talk about microtubule nucleation, meaning to say, how do the monomers of the microtubule organize themselves, connect themselves together. So here we can see a cell undergoing cell division. And you can see here the spindle fibers. They are actually the microtubules. And you can see here, color-coded in green, the microtubule organizing center or the centrosome containing the centrioles. Now, this is color-coded to reveal that there's another type of tubulin. Not just the alpha and the beta found in the microtubule, there's also a gamma tubulin, exclusively found only in the mTOCs, the organizing centers. Now here's an electron micrograph. If you can see clearly, we have here the portion where the mTOC is, and then these strands here are the microtubules. So the negative end of the microtubule, of course, is here. Then we have the protofilaments outlined in the microtubule. So the nucleation starts, of course, here at the bottom, where the gamma tubulin are found. Okay, so first, there's a protein that's holding the gamma tubulins. And this complex is known as the gamma tubulin ring complex, or the gamma Turk. So when the microtubule is to nucleate or to form, it's the alpha tubulin side of the dimer, which is the monomer, that's gonna stick to the gamma tubulin that we see here. And notice the shape here already is sort of asymmetrical and it's not balanced. So that is maintained all throughout the length of the microtubule. So notice the shape, it's like a coil, it's not balanced. So this portion is hidden in the pericentriolar material. So as we've seen a while ago, the centriole is surrounded by the PCM. So that's where the gamma tubulin ring complex is hidden, and that's where the nucleation for the microtubule starts, with the alpha tubulin first sticking and then the beta tubulin away from the gamma terc. So if we move upwards, we can see here, this is now the positive end. And we can see the new monomers, which are composed of the alpha and beta tubulins, extending the length of the microtubule, with the alpha sticking first. So the end will really be the positive end or the beta end. And take note, this formation is catalyzed by GT-PACE. Okay, so it's a GT-PACE enzyme that allows for this reaction, the lengthening of microtubules, to happen. So here's another view of the centrosome. You can see the centrosome here. You can see the centrioles inside, a pair of them. And you can see the pericentriolar material, this green translucent structure. And you can see outlined the gamma tubulin ring complexes, the gamma terc. So when the microtubules are nucleating, they will grow this way to the outside. So plant cells lack obvious microtubule organizing complexes. Their microtubules are arranged this way, as we have discussed in the previous video, that's perpendicular to the length of the plant cell. However, even if there is no obvious microtubule organizing center, even if there's no obvious center, there are actually spots along these microtubules that start the nucleation of new microtubules. And that's where the gamma tubulin are found in plant cells. So take note, the difference is in animal cells, the gamma tubulin are grouped together in the microtubule organizing center. But in plant cells, the gamma tubulin are spread along these microtubules. So this shows a sequence of a plant cell that's about to divide, perhaps in mitosis. And you can see that the microtubules themselves just rearrange themselves so that they themselves become the spindle fibers, as you can see here, that will pull the sister chromatids in opposite sides. And here they facilitate the division of the two newly formed nuclei for the daughter cells. And they also serve as a position for the formation of the cell plate to divide the two new daughter cells. So this is cytokinesis in plant cells. Now we can see in this electron micrograph that these are the microtubules of the plant cell. And here we can see, pointed by the arrow, a growing microtubule. Okay, this arrow on top. So here it's quite short. Here it's longer, and here it's the longest. And here it's not obvious that there is a special point here that could give rise to a new microtubule. Actually, that special point pointed by this arrow with a stem contains the gamma tubulin of the plant cell. So if we're going to illustrate it this way, you can see that the gamma tubulin complex is actually located along this original microtubule, and it can give rise to new microtubules. So in vitro, microtubules actually have a very unstable assembly. meaning to say, once they form, they could spontaneously just collapse, as we can see here. Okay, so they could just collapse right away. Even if they elongate, there will sort of be split ends on the positive end, so they will not be able to form properly. Okay, that's in vitro. So there's something lacking. So that model of the formation of microtubule is actually known as the dynamic instability model. So meaning to say, at the positive end, as the GTP dimers, the one that contains the beta and alpha tubulin, assembled together with the help of GTPase, okay, as you can see here, they close up to have the assembled microtubule as we see here. At some point, they're going to collapse catastrophically. So that's why this is known as the dynamic instability model. So there needs to be a support here on this end to stop this from happening. And what is that support? So in this micrograph, you can see that the tips of the microtubules are actually glowing green. That's because they contain a special type of protein, a microtubule associated protein called plus tip. Okay, so that's needed in order for the microtubule to prevent from collapsing again once they form. So the plus tips, as shown here, is just one among the many of the microtubule associated proteins that are shown here. So you can see here the kinesin 13 for the disassembly of microtubules. You can see here catenin, the one that cuts microtubules. You can see the maps, the ones that help the microtubule stay organized along the length. And of course, the tips, which prevent the end of the microtubule from splitting. So if we're going to illustrate the gamma terc, which is where the microtubule organizing center is located in this negative end, and the tip, which is a special type of microtubule associated protein, is at this positive end to prevent it from catastrophically collapsing again.